Welcome to the Multiple Sclerosis Virtual Grand Rounds Educational Series. I'm Dr. Rae-Grant, Staff Neurologist at the Mellen Center for Multiple Sclerosis at the Cleveland Clinic. I'm the moderator for this series. It gives me great pleasure to introduce our speaker for today, Dr. Bruce Trapp. Dr. Trapp is the chairman of the Department of Neurosciences at the Cleveland Clinic and professor in the Department of Molecular Medicine, Cleveland Lerner College of Medicine. He has received many awards nationally and internationally for his groundbreaking work on the pathology of multiple sclerosis. He has extensive NIH research funding for his work and has developed a large basic research group who continue to extend the boundaries of our understanding of MS and its pathology. Today he will give a talk entitled The Pathogenesis of Neurological Disability in Multiple Sclerosis.
Well, greetings. I'm Bruce Trapp from the Department of Neuroscience at the Lerner Research Institute in the Cleveland Clinic. And I'm going to tell you about the pathogenesis of neurological disability in multiple sclerosis. I'm going to start by making a few comments about the disease MS. It's an inflammatory disease of the central nervous system that destroys myelin, oligodendrocytes, and axons. And in this context of inflammatory disease, we mean the entry of large numbers of immune cells from the blood into the white matter of the brain. MS affects over 2.5 million individuals in North America and Europe, and it's the major cause of non-traumatic neurological disability in young adults. So with the exception of head trauma, MS disables more young adults than any other disease or condition. MS affects females 2 to 3 times more than males. A few comments on disease progression. The clinical course is variable. I like to refer to MS as a very misbehaved disease. It follows no rules or regulations. Second point is much of the disease is clinically silent. So this is a challenge for physicians, many which have to prescribe unpleasant treatments such as daily or weekly injections to young people who feel rather invulnerable to this disease. So it is a challenge. We don't fully understand the pathogenesis of MS, and we don't know whether it's a single disease or a common endpoint of multiple disease etiologies. Now, in the next slide, despite this variability in progression, the majority of MS patients, and that's about 80-85%, undergo a biphasic disease. And that is, they start initially with a rapid advancing clinical disability. This can affect any really part of the central nervous system, vision, sensory, motor, and this disability is rapid onset, and it lasts for a few weeks, and at most for a few months, and then the disability disappears, the patient gets better, and is in remission, again for a variable amount of time. That can be months, years, and even decades in rare occasions, but the majority of these MS patients will have another neurological attack, again, that lasts for a few weeks or a few months at most, and the patient gets better. So this relapsing remitting stage of the disease, again, can last for a year or for decades. Now, we have a pretty good, I think, take on the cause of this disability in the relapsing remitting MS disease. And the next slide shows some MRIs for a relapsing remitting MS patient's central nervous system. And usually when the patient comes in with a disability that is thought to be MS, the physician will order an MRI scan, and that MRI scan will have GAD enhancement for looking for breakdown of the blood-brain barrier. And on the panel A here shows an MRI showing lesions within the white matter. These occur as bright spots. On the right side is the GAD enhancement of that, so we see the 2 lesions or 3 lesions here are new lesions. So it is generally accepted in the MS field that these episodes of disability or relapses in the early stage are caused by white matter lesions. So what are the white matter lesions? Next slide will show you a cartoon of what goes on in these white matter lesions. And this is a slide of a human brain, stained human brain section, white matter section, stained for neurofilaments, the axons in red, and the myelin that surrounds those axons in green. And an early event in the MS lesion, as shown in this slide now, is the entry of immune cells from a blood vessel, and these immune cells certainly participate in the destruction of myelin around the axons. And we can tell a very active lesion in an MS brain by the deposition of the immune cells and the presence of myelin debris within the monocytes or the macrophages within the lesion. Now, in some of these lesions early in the disease process, I'm going to take away the immune cells. There can be a repair process, and that is that the lesion will generate new oligodendrocytes, as you see coming up here in green, and these oligodendrocytes can remyelinate the demyelinated axons. We can identify those pathologically at autopsy by the fact that the myelin is thinner than usual and the inner nodes are shorter. These are called shadow plaques. Generation of oligodendrocytes in the MS lesion is the best example of cell replacement in the adult human brain, and it can involve rather large areas of the CNS. Unfortunately, this repair process does not occur in all lesions, and in fact, the vast majority of lesions do not repair, and what happens is, as shown in the slide now, is that remyelination does not occur, but a astrocytic glial scar occurs. So these are the chronic MS lesions, and really it's these gliotic scars that helped with the name of multiple sclerosis. So this sort of summarizes the next slide of what happens. So you see in panel A, or neuron A here, is a neuron projecting to its target. And remember, this axon can be in the human more than a meter in length. And to make the action potential of the signal go quickly, evolution has devised the mechanism of insulating that axon, just like you would insulate an electric wire. And an early event in the MS lesion is shown in neuron B, is the invasion of immune cells from the blood into the white matter that demyelinate the axon. It's probably the edema that's associated with this influx of immune cells that caused the rapid onset disability. With time, that edema resolves and the patient begins to become better and neurological disability go away. There's two things, or really three things that contribute to that. That's the resolution of the edema. As shown in C, the demyelinated axons can redistribute sodium channels so the nerve impulse can get through the lesion, may slow a bit, but once it hits the other side, off it goes. And then, as I just demonstrated earlier, some of these lesions can remyelinate. That will also, this remyelination will contribute to the remission of the disability. So while that is a very good explanation of the disability associated with relapses and relapse and remaining MS, it is not a good explanation for the secondary progressive stage of MS. And this is when the point is reached where neurological disability is continuous and irreversible. And why doesn't inflammation account for that? Well, because we know many patients continue to decline neurologically in the secondary progressive stage of the disease when there is no evidence of new lesions as identified by MRI. There's no pleocytosis in the CSF. And most importantly, the patients don't respond to anti-inflammatory therapy. So there has to be something in addition to inflammatory demyelination that contributes to this continuous disability that we see in secondary progressive MS patients. And I think there is now, on the next slide, consensus that this is axonal loss in MS, and so I want to spend the remainder of the time talking about environments or conditions that cause axonal loss in MS. And the first is that axons are transected during inflammatory demyelination. And the next slide shows 3 panels of immunostained MS brain. The top left shows staining for the major myelin protein, proteolipid protein. So the black is normal myelinated cortex, and the white is the lesion. And this is a very active lesion, MS lesion, and we can tell that by the presence of immune cells. In panel B, this is a serial section stained for MHC class 2. So we know that this lesion is probably less than a few days old based on this. And you can see by the arrowheads in panel A some myelin debris that's still present in the immune myelin protein debris that's still present in the monocytes within that lesion. Now, panel C is showing at higher magnification than A and B, but it's a serial section to those. And the lesion border can be seen from the top right to the lower left. And this panel is stained for neurofilament protein. This is an axonal protein that we know this epitope is increased in demyelinated axons. And so when we first did this, I wasn't surprised to see an increase of this neurofilament staining in the lesion. What was surprising was the shape of the staining. And if you look, many of these structures look like ovoids. And in fact, that's what they are. They're ovoids described by Cahal about 100 years before we stained that slide. And what these ovoids represent are the cut ends of axons. And that's demonstrated in the next slide, which is a confocal image, putting these ovoids into 3 dimensions. So we take pictures of multiple slices of tissue. These have been compressed in this slide, but the point is, if you look at the arrows, you can see that there's an ovoid with one connection to it, and this represents the cut end of an axon. So we quantified these or counted these in various lesions and eliminated axons that just had a swelling. If you look at the arrowhead, in panel C here, you can see a swelling with 2 connections. So that's a swollen axon, but it's not a transected axon, and I'll emphasize the difference between those 2 in a minute. So what was the big surprise? The big surprise were the numbers. So if we look at these active lesions that have the immune cells filled within the lesion, probably only a few days old, we found that the number of transected axons was over 11,000 per millimeter cubed. Now, when the radiologists look at these lesions, they measure them in centimeters cubed, not millimeters, so you get some idea of the extensive axonal transection that occurs. If you look at chronic active lesions a little bit older that are growing at their edge, maybe expanding at the edge, you can see there's over 3,000 transected axons per millimeter cubed there. If we look at control white matter, there's less than 1. So here's the concept here, is if we look at that neuron here in panel A in the next slide, projecting an axon to a target, as inflammatory demyelination occurs, there's some collateral damage and transection of the demyelinated axons. And there's a big difference between a demyelinated axon and a transected axon. In the central nervous system, if you cut a long projection axon in the white matter, the loss of function, or the function of that neuron that extends that axon, is lost forever. So a demyelinated axon can retain its function either by redistributing sodium channels or by being remyelinated. A transected axon, the function of that neuron is lost forever in the central nervous system. Now, the next slide just shows that this axonal transection and ovoid formation can occur during active stages of myelination. So this is a confocal image of an MS lesion that's actively undergoing demyelination. You can see three major axons going from left to right stained in green. The red is the myelin surrounding that axons, and you can see between the arrowheads here that segments of these myelin sheets have been demyelinated. And if you look at the lower axon, you can see that ends in an axonal retraction bulb there indicating transection. So to summarize axonal transection in MS, the next slide, it begins at disease onset. So we had a patient, individual, in that study that had a 2-week disease duration, had active lesions, and all active lesions, as far as we have seen, have significant axonal transection. We believe the transection is related to the inflammatory activity of the lesion, so it's collateral damage, that is that the demyelinated axons are vulnerable to the inflammatory environment. At present, there's no evidence that there's a direct immune attack on axons. I don't think we can eliminate that possibility at this time. If it does happen, it would have to be an immune attack against a subpopulation of axons, because despite these high numbers that I've shown, the vast majority of axons in a demyelinated lesion survive. So let me change now to chronic demyelination results in axonal degeneration. And this is a hard thing to prove in the human brain, because we'd have to sample axonal numbers at different time points, and of course we don't have the ability to do that. So I'm going to present some data that supports this concept, and that is, as shown in A on the next slide, that many axons, demyelinated axons, survive the initial demyelinating event, redistribute sodium channels, and probably around for extended periods of time, years, decades in some cases. But that axon, that demyelinated axon, is at risk to degenerate because it loses trophic support from the myelin-forming cells. And that axon will eventually be transected, as shown in B, or the neuron will degenerate, as shown in C. So what's the proof of principle for that? And one of it comes from basic science studies that we and others have performed over the year that were designed to alter the protein composition of myelin, and really designed to look at the role of those proteins in myelination. And what we found in removing some of those proteins, and the first was MAG, myelin-associated glycoprotein, that there was no alteration in the myelinating process. But what we found is that with age, axons in the MAG-deficient mouse began to degenerate. So this was shown that MAG plays a role in providing this trophic support that's required for long-term survival of axons. In addition to MAG, there's been two other proteins removed from myelin, PLP and CNP, that also show a primary axonal pathology with little or minimal alterations in the myelin sheath. So the next slide shows another piece of data that supports degeneration of chronically demyelinated axons. So these are two brains from our rapid autopsy protocol, in which we perform MRIs on the brain in situ before we take it out. The left shows MS brain 18, and you see the typical white matter imaging changes, the bright spots that are primarily localized periventrically in this brain. Now, right below MS-18 MRI is a slice from that brain, and you could see what one would expect from that MRI, and that is that there is a huge periventricular lesion, as shown by the gray, around the ventricle within this brain. Now, if you look at MS-9 on the right, the MRI, this is one of the most atrophic brains I've ever seen at autopsy. This is an MS brain. There's very little white matter left, and what white matter there has abnormal MRI signal. If you look at the slice below, which is from that brain, it's quite shocking to see there are no space-occupying lesions within the white matter. the white matter is not normal. There are abnormalities in myelinated axons there, such as swelling of the myelin sheath, but the striking thing is there's no space-occupying lesions. The only interpretation I have for that is that all the demyelinated axons that compose the lesions have degenerated, and as they degenerate, the space-occupying component of them disappears. So here's the concept here. If we go to the next slide, is that demyelinated axons survive the initial stage or event of demyelination, redistribute their sodium channels, but with time they undergo chronic changes, and we and others have characterized these. I don't have time to summarize those right now, but I will at the end. What happens is the axons swell when they're demyelinated. There's dramatic changes in mitochondrial properties, endoplasmic reticulum, and eventually the axons degenerate. Now, how bad can that get? And that's a question that's often asked, is how much axonal loss do you get? It's very hard to do that in the brain, except for some select fiber tracts where you can get the dimensions of the fiber tract, because atrophy plays a significant role in quantifying the number of lost axons. We developed a method years ago to do that in spinal cord, where we got an estimate of axonal loss. And this is the data from that paper there, showing 2 levels of spinal cord in very disabled patients. So inclusion criteria here was quadriplegia, so they couldn't use either limb, upper or lower, and we wanted to know how bad could it get. So this is axonal loss within spinal cord lesions, and you can see the numbers there, 78, 72, 75, 62, etc. Average axonal loss in these regions was 68%. So they had almost lost 70% of their axons. That's why these patients were quadriplegic. It wasn't because their axons were demyelinated. It was because they had degenerated. So to summarize degeneration of chronically demyelinated axons in MS, we believe it's a major contributor to permanent neurological disability. It results from loss of myelin trophic support. It may not be the only thing, but certainly a major contributor. And we believe that axonal mitochondrial abnormalities cause loss of function, which is reduced ATP or energy production, and toxic gain of function, which is increased reactive oxygen species within the axons. So overall, when you demyelinate an axon, it increases the energy demands for nerve conduction, but the demyelination results in reduced ATP production, and that establishes a vicious cycle of increased axoplasmic calcium, reduced axonal transport, activation of proteolytic enzymes, and reactive oxygen species, destruction of cytoskeleton, loss of sodium, potassium, ATPase, and eventually axonal death. So let me now turn to the third area, and probably one of the hottest areas of MS research right now, which is cortical and gray matter demyelination. So the next slide shows a slice of an MS brain, and if you just look at brain slices, you can divide them into 2 parts, the white and the gray. And the white part is the white matter, where all the myelin fiber tracts are concentrated, and this part of the brain is white because of the myelin. Myelin's 80% lipid, and lipid is white. And if you demyelinate an axon, the white matter turns from white to gray. And you can see on the left side here a huge periventricular lesion that is gray due to demyelination. Now, the cortex also contains myelin, but it's not white because myelin makes up a much smaller component of the total tissue area. We have all the neurons, the synapses, the dendrites there. So if you demyelinate a cortex, it doesn't change colors. So you can't see cortical lesions macroscopically on a brain slice. We also do not see them routinely on MRI analysis, and I'll show you a reason for that in a minute. But what we do know now is that cortical demyelination and gray matter demyelination is quite extensive in MS brain. In the next slide on the top, you'll see 3 distributions of cortical demyelination in the MS brain. The first, in A, is the leukocortical lesion, and this is a lesion that's both in the white matter, subcortical white matter, and the cortex. the two are contiguous. I like to study these lesions because it's very likely that the white matter and cortical part of the lesion are of the same age. So when you see differences, you know they're due to differences in the environment of the demyelination, not to the age of lesions. Intercortical lesions are small areas of perivascular demyelination. We don't pay too much attention to those because they don't contribute significantly to the total lesion load. Probably the most fascinating lesion is the subpial lesion, and this is a band of demyelination that comes down from the surface of the brain and usually stops at layer 3 or 4, and these lesions can go over several gyri within the brain. Now, we know that cortical demyelination also causes neuritic transection of neurites and dendrites, as shown in panel E, and in chronic lesions there can be evidence of neuronal apoptosis, as shown in panel F. So I'm just going to show one of these lesions, the subpial lesions. So the next section shows staining of myelinated human cerebral cortex, and you see the dark black regions here. This is the white matter, and then between the dark black and the blue lines is the cortex, and you see that has a gray appearance. And so this is just showing that there's myelin throughout the cortex. It's just found at a less amount than found in the white matter. Now the next slide shows a section containing one of these subpial lesions in addition to a white matter lesion. So again, the dark black is staining for myelin within the white matter. You see a hole in the white matter tissue. That's not a real hole, that's a ventricle. And around that you have light gray appearance, which is a white matter lesion. So if you then go from the white matter into the cortex, you see a dark gray, that's myelinated cortex, and then outside to the blue line you see a subpial lesion going over several gyri here. If you just look at this slide and try to do a semi-quantification at macroscopic view and ask yourself, is the gray matter lesion load or the white matter lesion load greater, you would come out with the answer that the gray matter lesion load is greater. So let me show an active type 1 lesion here and show some differences. So this is a section from MS brain. You see a lesion. Again, it's been stained for proteolipid protein, the major structural protein of myelin. And I'm just going to use a green line to demarcate the white matter and the gray matter. And you can see the lesion is both in the cortex and in the subcortical white matter. And I'm going to show now there's the cortex in red, there's the white matter in yellow lesion. And I'm going to show the next section stained for MHC class 2 to stain these immune cells. And here you can see intensive staining of the white matter portion of the lesion, but not the gray matter, except for at the lesion edge. So if you look at this line on the MHC class 2 stained section on the right, it sort of shows the edge of the cortical lesion. These are activated microglia at the edge. So what one can see is this huge difference in the number of immune cells between the white matter and the gray matter. There is an increase in the number of immune cells in the gray matter if we go down and count, and that was published in the Peterson paper. But the increase is very small compared to the increase that occurs in the white matter. Now, I mentioned that we don't see these lesions by MRI, and most of our standard MRI techniques measures changes in water, and certainly edema being one of them. If you look at the next slide, it's a high magnification of one of these leukocortical lesions. So the cortex up and top is demyelinated. You can see the difference in the density of the demyelinated regions between cortex and subcortical white matter in the center here. And the slide to the right has been stained for serum proteins. And what you see is a remarkable accumulation of serum proteins in the white matter lesion, but not in the gray matter lesion. So there's very little breakdown of the blood-brain barrier in these cortical lesions, no increase in edema, and therefore no GAD enhancement or detection by standard MRI methods. So cortical demyelination, to summarize, it's present in most MS brains. It may be more prominent than white matter demyelination. We don't know for sure because we have no really good means to measure it globally. It contains few inflammatory infiltrates and no breakdown of the blood-brain barrier. The leukocortical lesions, the cortical part of leukocortical lesions, do have an increase in immune cells. As far as we know, the vast majority of subpeel lesions do not have increase in immune cells from the blood. Cortical demyelination causes neuritic transection in neuronal death, and one would argue that this must contribute to neurological disability. And at present, there's an urgent need for non-invasive methods to detect cortical MS lesions so we can really understand the dynamics of how they form and how they correlate with disabilities. Our current MRI methods are missing a major part of the pathogenesis of MS by not having the ability to identify these lesions. So gray matter pathology. Let me just make a few comments about that, in that gray matter atrophy is a significant and early aspect of MS pathogenesis and one of the best known predictors of disease progression. So one of the most important questions in MS research, in my mind, is whether gray matter atrophy occurs independent of white matter and gray matter demyelination. We don't know the answer to that, but there's a few clues that I just want to mention. And so ask the question, next slide, is gray matter atrophy a consequence of white matter demyelination? Axonal transection in white matter lesions will cause degeneration of synaptic endings and degeneration of neurons. So if you transect an axonal lesion, the distal part of that axon going to make a synapse on the target will degenerate. On the other hand, there's a lot of evidence that suggests that gray matter atrophy can precede significant white matter lesion appearances. And finally, cortices with long projection axons have less atrophy than cortices with cortico-cortical connections. This would support the concept that maybe white matter demyelination doesn't play a significant role in cortical atrophy, although it's just suggestive, it doesn't prove that. So the second question would be, is gray matter atrophy a consequence of gray matter demyelination? And we don't know that either because we don't have a reliable MR method to detect cortical demyelination in MS brains. There is data, however, that suggests that cortical atrophy is an early event in relapsing remitting MS patients when subpeel lesions are rarely detected. So to conclude, permanent neurological disability in MS is caused by axonal transection during inflammatory demyelination, degeneration of chronically demyelinated axons, and cortical pathology including demyelination and possibly a neurodegenerative component. And I think one of the major challenges for us in the MS field now is to try to understand the mechanisms of cortical demyelination, particularly in the subpeel lesions. So that's the end of my lecture, and thank you for being attentive.
Thanks, Dr. Trapp, for your up-to-date and comprehensive discussion of the pathogenesis of neurological disability in multiple sclerosis. It's astonishing to consider how much has changed about our understanding of MS and its pathology, thanks in no small part to Dr. Trapp and his colleagues. We used to think MS was all about demyelination, and now we know that axons are crucial to the development of disability and are a core part of early damage. We used to think that MS was a white matter disease, but we now can see that the gray matter and cerebral atrophy in general are critical to the development of disability in this disorder. I think we now realize that MS is a whole brain disorder, and one where ongoing injury culminates in disability, likely many years after initial pathological changes. Dr. Trapp's work encourages us to continue to revise our concepts of MS and to continue to question traditional ideas about this disease. Please remember to fill out your evaluation for CME credit. We look forward to having you participate in other parts of our multiple sclerosis virtual grand rounds.